I will present to you my work on flavonoid metabolism in Tetrastigma hemsleyanum diels at gilg based on metabolome analysis and transcriptome sequencing. I will lay my stress on the following 5 aspects. The first part concerns background. The second part concerns experimental design. The third part concerns results. First part concerns conclusion, and the last part relates to future prospects. To begin with, I would like to introduce you to the experimental material. Tetrastigma hemsleyanum diels at gilg is a rare Chinese herbal medicine, which is one of the new 8 flavors of Zhejiang. It is known as a plant antibiotic and possesses attractive effects, including anti-inflammatory, anti-tumor, and antioxidant. It is mainly produced in Zhejiang province and is widely distributed in Hangzhou, Jinhua, Lishui, Taizhou, and Ningbo, among other regions. Standards of processing Chinese food drugs record that the YouTubers can be used as medicine. Therefore, leaves are always regarded as the garbage, which causes a waste of resources in the process of medicine. Additionally, Tetrastigma hemsleyanum has been exhaustively developed for the multi-application of its YouTuber, which has led to a sharp decline in its natural resources. Further, its chaotic quality management has resulted in medicine being characterized by inconsistent future effects, and its efficacy is attributed mainly from flavonoids. However, the flavonoid composition and the expression of flavonoid-related genes are rarely studied in leaves and YouTubers of Tetrastigma. So, how do flavonoids accumulate in Tetrastigma? Using transcriptomics and metabolomics techniques, we collected YouTubers and leaves to analyze flavonoid components, flavonoid-related genes, and differentially expressed genes. We used the Pearson correlation analysis to find key flavonoid synthesis genes and validated them by quantitative polymerase chain reaction to determine relative expression. At the same time, we performed an enrichment analysis of differentially expressed genes. Through the above experimental design, we have obtained the following results. From Figure 1, we can clearly see that the accumulation of flavonoid monomers was obviously different in leaves and YouTubers. By cluster analysis, we have found a good biological reproducibility in root and leaf groups, and the expression levels of camptophore, iridiopteryl, acetylcholine, epigene, and isocyanates were clustered together. As determined by GCMAS, we found that the concentration of phytosine, ornithine, meridine, aspartame in the leaves were higher than those in the YouTubers. Acetylcholine concentration in YouTubers was higher than that in leaves. Also, we found that luteolin, luteolizide, hexbridine, lutein, lomethrin, bacillin, and epigene 7 oxygen glycoside were found to only be contained in the leaves. These ingredients, most of these ingredients has the effect of anti-inflammatory and anti-tumor. So we think that the leaves should also be used. It should not be discarded. Additionally, Tetrastigma has a variety of flavonoids. According to flavonoid basic skeleton, we have divided the detected monomers into 6 classes, including flavones, flavone glycides, flavonols, flavonoglycides, flavones, and flavonoglycides. Then I shall now switch over to the result of transcriptome sequencing of leaves and YouTubers. To understand the relevant biological significance, the assembled imaging searched against 5 databases were annotated, including KOG, NR, SweetProt, KGD, and GO database. In this study, we found that 31,820 functional genes were annotated. 28,757 were found in leaves. 33,053 were observed in the YouTubers. Next, the assembled imagings were statistically annotated in 3 biological functions in GO database. The study showed that 22 groups in biological processes, 25 and 3 in cellular components, 303 in molecular functions. The imagings that matched with the KGD database were divided into 5 branches. From Figure 7, 1,751 genes were related to secondary metabolism. 2,517 genes were related to environmental adaptation. So the further study on differential expressed genes of leaves and YouTubers, a result similar to the differential metabolite analysis emerged. About 67,345 DGs were identified between leaves and YouTubers, including 6,876 upregulated genes, 14,277 downregulated genes, and 46,192 non-DGs. Immediately from this, we performed an KGD classification of differentially expressed genes. We found the majority of DGs were obviously enriched in the metabolism pathway. The top 10 significant pathways were listed in Figure 10. Surprisingly, the DGs were obviously enriched in photosynthesis and China protein pathway. From Figure 11, a total of 25 DGs belonging to the chlorophyll A over B blending protein family was enriched in the photosynthesis and China protein pathway. Now I'd like to give a brief introduction of photosynthesis and China proteins. Many researchers showed that about 95% of plant biomass accumulation is attributed to photosynthesis, and photosynthesis and China proteins can regulate the ability to capture solar energy and transfer the energy to reaction center. It can also affect the synthesis of carbon skeleton that makes up the flavonoid compounds. So this study indicated that the photosynthesis and China proteins may be key regulatory proteins that can affect the accumulation of flavonoids. Moreover, our team previously performed a different shading experiment on Tetrastigma, and we found that light intensity can affect the accumulation of flavonoid. The next reflection is about a correlation between flavonoid-related genes and flavonoid content. The biosynthetic pathway of flavonoid is listed in Figure 12. It contains 11 key genes and 22 metabolites. The heat map of synthesis expressions reveal that the expression levels of CHI UIGT in the leaves were higher than those in the roots, and the expression levels of PAL, CHS, F3H, and FNS in the roots were higher than those in the leaves. to reveal the relationship between the key gene and the flavonoid content, we perform a Pearson correlation analysis. In figure 13, the epidemic 7 oxygen glycoside quercetin, erythritol, luteolin, acid quercetin, asparagine, cryptoeryl, and naridine were highly correlated with the gene expression of CHI. And we found vitacin, ornithine, lanocerin, lutein, isoquercetin, luteoliside, luteolin, asparagine, naridine, erythritol, myristotin, biclin, and cryptoeryl, dielsmetin, and quercetin were highly correlated with the gene expression of UFGT. The CHI is the second rate-limiting enzyme involved in biosynthetic pathway of flavonoids. It can catalyze the intramolecular cyclization reaction and transfer bicyclic calcium into cyclic flavonoids. UFGT is a downstream enzyme which is generally considered a key gene, which is related to the synthesis of hemsleyanum. However, some researchers also indicated that UFGT play an important part in isoflavonoid synthesis. We think that the catalytic rate of the enzyme and the consumption rate of the substrate may lead to this phenomenon. Next, we perform a quantitative polymerase gene reaction to determine the differentially expressed genes. The cluster plot of the correlation between iron-6 and relative gene expression indicated a positive correlation. This result verified that the iron-6 data is reliable. To further validate these two key genes, we collected Tetrastigma specimen from Hangzhou, Jinhua, Lishui, and Taizhou to analyze the relative expression and the total flavonoids. From figure 15, key genes' relative expressions in leaves and root tubers showed similar trends to those obtained from the iron-6 data. And from figure 16, the concentration of total flavonoid in the leaves were significantly higher than those in the root tubers. The leaves and root tubers of Lishui Tetrastigma were of a better quality than the others, followed by Taizhou. In conclusion, I would like to indicate the following 3 points. Firstly, given that the active ingredients of flavonoid in leaves are higher than those in the root tubers, we suggest that leaves should also be utilized. Secondly, we found a new key regulatory protein called photosynthesis antenna proteins that play a key role in flavonoid biosynthesis pathway of Tetrastigma. Lastly, CHI and UFGT were the critical genes of most flavonoid metabolism in Tetrastigma. Nowadays, toothpaste and diels made from Tetrastigma root tubers were available in supermarkets. Based on our study, we think that leaves may also be divided into daily chemicals to prevent waste of resources. By the way, in folklore, leaves are used against shingles and mouth ulcers. After using it personally, we found it do have a good effect. As a consequence, in the next step, we will perform an in-depth study on Tetrastigma to address these two healing effects. Finally, please allow me to introduce our team. I'm a member of Forestry Medicinal Herbal Resources Mining and Quality Control Team. Our team focused on the development and utilization of traditional Chinese medicine resources, cultivation and physiological of medicinal plants research. And the lady in the middle is my tutor, Professor Yan Bai. That brings us to the end of my presentation. Thank you for your attention.